So tell me a bit about what the poster is about. What was the paper originally about? What were they trying to achieve?
They were just trying to create some new dyes, and when they created them, they found out that they had special molecular properties, so that they could target mitochondria, and then they fluoresce in that, which could make them interesting.
So the idea is that by the dyes targeting the mitochondria, you can see when the mitochondria are active, or you can see where they are. Is that the idea?
They can be used as biological trackers. So I think then they may have some applications in medicine if you did some further research. But they were just seeing if it was safe to start with. So they saw that there weren't any changes to the cell, except that you could see the fluorescence, so you could use them as trackers.
Right. So you could put these into the cell, you can see the fluorescence, it's not causing any change to the properties of the cell. Perfect, yes. So what is special about the molecules? Now, you know me, anything bigger than methanol is too complicated. So what's special about these molecules?
When they're put in acids, they become mono-ring opened anionic semi-naphtha rhodofluorescents, but when they're put in base, they become ring opened again, but they're still rhodofluorescent, and there's also ring opening when they're in mercury. So you can put them in any environment and they still become ring opened and they still become fluorescent.
Right. Okay. So is this the sort of area that you would like to go into when you come to do your research projects next year or the year after? Or have you looked at this and now you're thinking perhaps you should do something in physical chemistry, perhaps?
For me, I want to do organic chemistry, but I want to look into kind of more cosmetics and the way that the fats and lipids can interact with different things.
I'd rather go into physical chemistry. The sort of aspects of the maths is a bit easier than the mechanisms of making things like this. So.
Yeah, well, I'm with you all the way, of course.
I don't think I would go into organic chemistry either.
So this poster has been really useful in helping you decide the future course of your degrees, yeah? Fantastic. Thank you very much.
Our poster is on a research paper that is to do finding out the oxidative steps for the biosynthesis of scolestatin. And the reason this is useful is that it can be used in, there's a potential use in the control of cholesterol biosynthesis. So these are the oxidative steps that they didn't understand before. And they found out that there are 3 enzymes that are involved in this oxidative synthesis. The MFR1, the MFR2, and the MFM1. They did this using gene knockout experiments using those graphs. So this graph is MFM1 without the MFR1 and MFR2, and what they found was that the oxidation happened early on. So yeah, they use gene knockout, and they also use heterologous using heterologous expression analysis. So they put benzo coenzyme into the ALZ to figure out how the MFR1 enzymes and the MFA and all the other enzymes work together, and see what they produced to figure out what order they happen in. And then the order's sort of shown there, and this is the whole reaction pathway.
So what techniques were they using for producing these knockouts?
The knockouts?
How did they make those?
The knockout experiments is that they basically deleted genes or deactivated genes, and showed how the reaction would work if those enzymes were present or weren't produced.
In this proposed reaction mechanism, at this step, can you tell me how many NADPH molecules are involved in this reaction?
Yes, so you would only use one because this proton here would be attached to this oxygen here, and then that would cause the reduction, and then the electrons migrate to here, and so only one molecule would be used to reduce in here.
Okay.
So it would be bioactivated. That's why we use beta-lactone, bioactivated by this enzyme, and it will produce the hydroquinone, which will eventually cause cancer cell death by attaching allosterically to 5-law.
Okay.
The good thing about this redox cycle is that it will also generate raw species, so reactive oxygen species like superoxides, which also cause cancer cell death by breaking the DNA strands.
Okay.
So this is the proposed reaction mechanism.
So this part of you're talking about the binding model, right?
Yeah.
Of this molecule.
Yes.
So can you simply describe what is going on here?
Yes, so basically you're using a mutant or like the aberrant expression of 5-law that you would basically see in the neutrophils in the white blood cells when they're mutated. Then we're going to construct the homology model, which is like an atomic resolution model to be used in our software.
So is this a crystal structure or is it solved by crystallography?
It could be used by crystallography. In this experiment, what they did is actually get data from different data sets and build their own machine intelligence to be able to create the homology model. So I'm not sure if they did crystallography.
I can see actually, so you are using this homology model, right?
Yeah.
And they try to predict the binding pocket.
Yeah.
And then you have your
So we
the
get
molecule.
beta-lactone
Yeah.
and we dock it to the predicted allosteric site. And once beta-lactone is docked into this protein domain interface, then it will bind hydrogen bonds in these 3 sites and then an amide pi stacking in Q12.
Yeah.
And that's how we'll basically do its anti-cancer activity when binding to 5-law.
Okay, thank you, very nice.
So our poster was based on a paper about an organoplatinum metal cage used as a multi-modality thermostatic platform for cancer treatment. So it's a new combination of previous cancer treatment techniques sort of rolled into one, as well as it works an awful lot better than previous cancer treatment techniques as well. Being able to kill off tumours completely and the only treatment so far to not have give a recurrence of a tumour. So yeah, it's a very impressive treatment.
Okay, so when you say photochemotherapy, what does that mean?
So, it oxidizes oxygen from a triplet to a singlet spin state. And this has an oxidative effect on the tumour that is being treated, which can kill off the tumour cells.
So that's the photodynamic therapy, is this photosensitizer, and it absorbs light and then it irradiates the energy and excites molecular oxygen to singlet state oxygen, which forms oxidative damage to tumours, and then it's bound to a CPT unit, which is a chemotherapeutic drug, and that kills off all the residual tumour cells.
So does this only work on tumours that are close to the surface of the body if you need to irradiate them to get this to work?
No, because there's a targeting ligand, and then that goes towards the tumour and drops off the nanoparticle.
Okay, so do you want to tell us a little bit about your poster, Pal?
Our poster focuses on near-infrared light-triggered drug release from the silicon nanoparticles. There are 2 types, the near-infrared thermal conversion and near-infrared light into UV. And I'll briefly describe the UV section. Basically, the drug is carried by a carrier, which is usually a lanthanide ion dope material. Which the good thing about a lanthanide ion is that not only does it undergo down conversion, but also up conversion. An up conversion is basically when 2 or more photons of lower energy, in this case near infrared, are excited to an upper level energy and releases a 1 photon UV light. Now this photon then, on the diagram, splits the drug from the carrier, which the drug is then at the tumor site, attacks the tumor on the tumor site. The benefits of using near infrared is that there's deep tissue penetration, minimal autofluorescence, and high biosafety, which basically means it's safe to use compared to, let's say, UV, which has very low tissue penetration and quite low biosafety, but in this case, it's almost as if applying the UV radiation at the tumor site, which releases the drug.
Certain molecules have the ability to convert the NIR light into thermal energy, such as gold nanorods, and we can see the example here. This is the gold nanorods, and it consists of a NIR thermal converting coil inside the gold nanorods, and a layer of mesoporous silicate coated with it, and there's thermal molecules functioning as a gatekeepers, and when NIR radiation produced into the molecule, and the heat generated, and the driving force can be used to accelerate to diffuse the drug within the mesospores of MSNs for controllable drug release. And here is the diagram. And NIR radiation beam into it and drug release.
So there's a few issues with the research, and that's what the future advances are going to look at. One is that the nanoparticles aren't able to contain more than one drug without mixing them, which is obviously doesn't work. So it's important that the MSNs, the mesoporous silica nanoparticles, are able to interact with multiple wavelengths so that they can release different drugs at specific times depending on the wavelength of the NII light that they are exposed to. Also, the storage within the MSNs is also going to be looked at, and we're looking for an MSN that has multiple storage compartments. And another development that's being looked into is being able to follow the drug after it's been released within the body. So those are a few future advances.
So could you explain to me what your poster's about and the name of the poster, please?
So the name of the poster is Catalytic Farming, Reaction Rotation Extends Catalyst Performance. And what we see here is how this catalyst can be regenerated by using different kinds of reactions. So for Sonogashira coupling, the catalyst is depleted, between commas, as it's being kind of consumed, and that affects the yield. If we mix with other reactions that can regenerate it, such as Ullmann homocoupling, then we can restore the good functioning of the catalyst. So these are some cycles that have been found, and it's basically the same idea as crop rotation in farming, where we change crops every year so that the soil doesn't get depleted. This is the same idea, so that the catalyst doesn't get consumed. We just change the reaction every cycle so that the catalyst is being regenerated again, and that way we never run out of catalyst, which is what the problem reaction makes. It makes it go to palladium oxide instead of being nanoparticles of palladium. And yeah, these are the combinations of reactions and the yields that have been obtained. So by mixing them, we can make the problem reaction have 6 cycles and still get an 80% yield. Whereas with just the problem reaction, there's 3 cycles and you only get a 39% yield.